Hello guys, so today we are going to see antibacterial drugs. Okay, so the drugs which can kill the bacteria. Okay, antibacterial drugs. So generally this antibacterial drug can be classified in different ways. So suppose it can be classified as broad spectrum or narrow spectrum, or suppose broad spectrum versus narrow spectrum. If antibiotic is effective against many bacteria, then it is known as a broad spectrum. And if the antibiotic is effective against a few species of bacteria, then it is narrow spectrum antibiotics. Okay, so this is one way of classification. And there are another way like bacteriostatic, bacteriostatic versus bactericidal. Okay, so that means bacteriostatic means if the antibiotic only stops the multiplication of the antibiotic bacteria, then it is known as bacteriostatic, like your sulfonamides. Okay, already I have taught you the sulfonamides. Next is bactericidal. If the antibiotic kills the bacteria, then it is known as bactericidals. Okay, so these are some ways through which we can classify the antibacterial drugs. And generally the selection of antibacterial drugs depends on the person, depends on the species of the bacteria, bacterial infection. Okay, also, and the method which determines the selection of antibacterial drug is commonly known as Kirby Kirby-Brewer method. BA Brewer method. Okay. So this method is generally used for the selection of antibiotic. Okay, used for the selection of antibiotics. Okay, so let's move on to the different mechanisms through which antibacterial drugs act on the bacteria. So if you can see in the slide, there are different ways through which antibacterial agent act on the bacteria. Suppose they may act through disruption of DNA, like nitroimidazole and nitrofurantoin. They may inhibit the topoisomerase enzymes, okay, like quinolones used to do, and they may inhibit the folate synthase, like sulfonamides and trimethoprim. This is already over for us. And then they can inhibit the protein synthesis at various sites, like macrolides, aminoglycosides, tetracycline, tigecycline, chloramphenicol, glycosinamide, fusidic acid. Okay, so this all will inhibit the bacterial protein synthesis, and some may inhibit the cell membrane synthesis, and they will disrupt the cell membrane. Okay, they will disrupt the cell membrane, like polymyxin and daptomycin. Okay, daptomycin. Next is your cell wall peptidoglycan disruption, means cell wall inhibitor. Okay, like penicillin, cephalosporin, monobactam, carbapenem, and glycopeptide. So these are the different mechanisms through which antibacterial agents act on the bacteria. Okay, so let's firstly move on to the first topic. So today we will be discussing about just introduction on the cell wall inhibitors. Okay. So before discussing on the antibiotic which act on the cell wall of the bacteria, let's see the makeup of the cell wall of the bacteria. Okay, so bacteria are generally classified into gram positive and gram negative. So gram positive have has a very thick layer of peptidoglycan. Okay, gram positive has a very thick layer of peptidoglycan, whereas gram negative, the peptidoglycan layer is very thin. Okay, here it is very thin layer of peptidoglycan. This is very thick layer of peptidoglycan. And another thing, another important thing to remember is in gram negative, this peptidoglycan layer is buried inside the lipopolysaccharide. Okay, this is lipopolysaccharide. So many antibiotic cannot cross this lipopolysaccharide, so they will be ineffective against gram negative, such as penicillin group of your antibiotic cannot cross through this lipopolysaccharide, so it will be ineffective against gram negative. But it is effective against the gram positive because it can easily access this peptidoglycan layer. Okay, so this is one important thing to remember. Now the next thing you have to remember that this gram positive, while doing staining like crystal violet, okay, crystal violet, if we do a staining of crystal violet, this gram positive bacteria will show violet color. Okay, violet color, it will show violet color, it will show violet color stain. Okay, because this violet, crystal violet, this crystal violet will be absorbed by this peptidoglycan layer, it will be retained by this peptidoglycan layer after washing also. Okay, after washing also this violet color crystal violet will be retained in the peptidoglycan layer. But whereas in case of gram negative bacteria, since the peptidoglycan layer is very thin, the crystal violet won't be retained so much inside the this thin layer. So when gram staining is done, when crystal violet is when gram staining is done using crystal violet, so the color of this gram negative bacteria will be your red. Okay, red to pink color, red or pink color, red or pink color. Okay, here it was violet, here it was violet or purple color, here it is red and red or pink color. So this is the difference between the gram positive and the gram negative, and the structural difference also you can see. Example of gram positive bacteria is clostridium actinomyces, mycobacterium, nocardia, streptococci, staphylococci. An example of gram negative bacteria are vibrio, campylobacter, pseudomonas, and members of the family of enterobacteriaceae. Okay, so let's move on to the next slide. So cell wall inhibitors, these are also commonly known as beta-lactam antibacterial agent or beta-lactam antibiotics. There are five class of drugs in this beta-lactam antibiotic. First is your penicillin, okay, then cephalosporin, then carbapenems, monobactam, and clavulanic acid. Okay, and this ring, okay, this ring is known as beta lactam ring. Okay, this ring is known as beta lactam ring. You can see everywhere it is present. Okay, this beta lactam ring is present everywhere. Okay, on all these five class of drug, beta lactam rings are present. So that's why it is known as beta lactam antibiotic. And this beta lactam ring is very important for their antibacterial property. Okay, so let's move on to the first class of drug, that is penicillin. Okay, first class of drug, what is that? Penicillin. So before going on to the penicillin, let's see some of the examples of your different type of beta lactam antibiotic. For penicillin, you can see penicillin G, penicillin V, nefcicillin, oxacillin, diclofenac, amoxicillin, ampicillin, piperacillin. Then for cephalosporin, there will be first generation, second generation, third generation, fourth generation, and advanced, that is fifth generation. And for monobactam, example is H3onam, and for carbapenem, it is example meropenem, doripenem, atrapenem. So many are there. Okay, so the list goes on. Okay, so let's see. So now let's move on to the mechanism of action of beta lactam antibiotics. Okay, like penicillin. Okay, so what happens here is this round you can see these are peptidoglycan. Okay, these are peptidoglycan, and these peptidoglycans are made up of NAM and NAG monomers. Okay, NAM and NAG monomers. Okay, this round you can see first one is NAM, then NAG, then NAM, then NAG, then NAM. So one by one, NAM, NAG, NAM, NAG, NAM, NAG. Okay, NAM means N-acetyl muramic acid, N-acetyl muramic acid, and NAG is N-acetyl glycosamine. Okay, N-acetyl glycosamine and N-acetyl muramic acid. So this is alternate monomers of NAM and NAG. So this combined together they are known as polypeptide chain. Okay, so this polypeptide chain will be cross linked here. You can see a bridge here like this is cross linking. Okay, this is known as cross linking, and this cross linking is done by penicillin binding protein. Okay, this green color is penicillin binding protein. This penicillin binding protein helps in the cross linking of this peptide. Okay, polypeptide. Okay, transpeptide, you can tell transpeptide. Okay, so these transpeptides will be cross linked by the penicillin binding protein. Okay, again here it will be cross linked by penicillin binding protein. Again here it will be cross linked by penicillin binding protein. Again here it will be cross linked, then cross link. So in this way cell wall will be intact. Okay, the cell wall of the bacteria will be intact. Okay, if this penicillin binding protein is not there, there won't be any cross linking. Okay, and if there is no cross linking, the cell wall won't be synthesized properly. Okay, this cross linking process is known as transpeptidation. Okay, transpeptidation. Okay, transpeptidation, this process of cross linking is known as transpeptidation. What happens, all the beta lactam antibiotics will inhibit or bind to this penicillin binding protein. Okay, this penicillin binding protein, and it will inhibit the penicillin binding protein, and therefore there won't be any cross linking, and there will be so much gaps will be there in the your cell wall of the bacteria. Okay, so cell wall won't be synthesized properly, and through these gaps this all solids will from outside it will come inside through osmosis process. Okay, osmosis process, all the solids, all the solvent molecules will come from outside to the inside of this bacterial cell by osmosis, and due to that this bacteria will bulge. Okay, it will swell. Okay, and swell and it will rupture. Okay, this bacteria will swell and then it will rupture. Okay, so this is the process of what your beta lactam antibacterial agent mechanism. Okay, now let's move on to the classification of penicillin. Okay, classification of the penicillin. So penicillin is generally classified into three categories: natural penicillin, semi-synthetic, and beta lactamase inhibitor. Okay, so benzyl penicillin is an example of natural penicillin. It is also known as penicillin G. Then clavulanic acid, sulbactam, and tazobactam is an example of beta lactamase inhibitor. This semi-synthetic penicillin is divided broadly into three categories: acid resistant alternate to penicillin G, that is example is penicillin V, extended spectrum penicillin, and penicillinase resistance penicillin. For example, for penicillinase resistance penicillin is methicillin, cloxacillin, and diclocillin. For extended spectrum penicillin, it is divided into three parts: aminopenicillin, uridopenicillin, and carboxypenicillin. The example for aminopenicillin is ampicillin, vacampicillin, amoxicillin. For uridopenicillin, it is piperacillin, megalocillin, and for carboxypenicillin, it is carbenicillin. So this is the classification for the penicillin group. Okay, penicillin group. So in next class we will be going in detail on how in what basis this penicillin classification has been done, and what is the particular antibacterial spectrum, side effects, everything we will be discussing on the next class. So for today that's all. So please try to go through my video and prepare notes by yourself, my dear students. Thank you very much for listening to me patiently.